Meiosis by Rachel Stone, Jordan Stallings, and Sophie Eubanks. What is meiosis? It's the process of a diploid cell dividing twice to produce 4 cells with half the original number of chromosomes. Why is it necessary? Meiosis is necessary because it's for sexual reproduction and genetic variation. Diploid. A diploid is nuclei that has pairs of homologous chromosomes. Haploid is nuclei that has 1 chromosome of each pair. Bivalent is when homologous chromosomes pair together, and a homologous chromosome is a pair of homologous chromosomes which contains 4 chromatids and is sometimes called a tetrad. Synapsis is a pair of homologous chromosomes that are bivalent in the pairing process. Oh my god, what? I can't.
No, keep going.
Okay. Crossing over occurs at random positions anywhere along the chromosomes. Chiasmata, it intentionally holds together the 2 chromosomes in each bivalent, and random orientation is the process that generates genetic variation among genes that are on different chromosome types. Interphase is the phase of growing that the cell is usually in. In interphase, there are chromosomes with centrioles. This is the plasma membrane. The cytoplasm is the area within it. The nuclear envelope encompasses the chromatin, which is inside of it. Prophase 1. During prophase 1, differences from mitosis begin to appear. The chromosomes begin to condense, and they also pair up. Each chromosome carefully aligns with its homolog partner so that the 2 match up at corresponding positions along their full length. So these are the centrioles, the spindle fibers between the centrioles. The chiasmata is holding the 2 homologous pairs together, and then these are the homologous chromosomes and the nuclear envelope encompassing them. The nuclear envelope also breaks down during this process. Metaphase, the chromosomes move to the center. Homologous pairs line up at the metaphase plate for separation. When the homologous pairs line up at the metaphase plate, the orientation of each pair is random. So here are the centrioles, and then the microtubule, the metaphase plate, which is in the middle of the homologous chromosomes, and then the centromeres are still in between each pair. For anaphase, it's when the homologous chromosomes move to the opposite of the cell. So these are the centrioles. The sister chromatids are on the end of the homologous pairs, and then they're splitting apart. In telophase, the chromosomes arrive at the opposite poles of the cell. In some organisms, the nuclear membrane reforms and the chromosomes decondense. So here's the cleavage furrow between the 2 cells, and then they've separated with the nuclear envelope around each one, and then these are the centrioles. During prophase 2, chromosomes condense and the nuclear envelope breaks down, if needed. The centromeres move apart, the spindle forms between them, and the spindle microtubules begin to capture chromosomes. So now there are 2 separate cells, and then this is the microtubule right here, the centrioles, and then the dissolving nuclear envelope that's around the homologous pairs. In metaphase 2, the chromosomes line up at the equator. The 2 sister chromatids of each chromosome are captured by microtubules from opposite spindle poles. In metaphase 2, the chromosomes line up individually along the metaphase plate. And the centrioles are still right here. In anaphase 2, the sister chromatids separate and are pulled towards opposite poles of the cell. So the centrioles are here, and then the sister chromatids are separating right here. In telophase 2, the nuclear membranes form around each set of chromosomes, and the chromosomes decondense. The cleavage furrow is coming out, and the centrioles are here, and the nuclear envelope is still surrounding the homologous pairs. During cytokinesis, a nuclear envelope forms around each set of chromosomes. The cytoplasm divides. The chromatin, which is condensed, and the nuclear envelope, which is surrounding the chromatin. And then the centrioles are still along the outside of the cell. What is the product of meiosis? The products of meiosis are 4 haploid genetically different gamete cells. And there's 4 of them. And then our references.